Okay. All right. So we're going to see what happens, how more egg responds to sperm. Okay. Now, just beneath the plasma membrane especially, there's a very dense network of actin microfilaments, or F-actin. Now, you find microfilaments all over the cell, but just beneath the plasma membrane, it's especially dense. In fact, we often call that region about a tenth of a micrometer or so beneath the plasma membrane. It's often called the cell cortex. And it's especially rich in microfilaments, a three-dimensional network of microfilaments, and various proteins that are going to be regulating the organization of these guys. It's so dense, in fact, that very few organelles are found in it. The cortical vesicles are a little bit unusual in that they're kind of put there early enough in development before you get the full extensive cytoskeletal network. But for the most part, things like mitochondria and ER and stuff like that are excluded from that area because it's just, you know, it's like trying to build a house in a pile of brambles. It's not going to work very well unless you somehow clear those guys out. Okay, now what's going to happen is we're going to start getting microfilament elongation and joining individual microfilaments together to make bundles of microfilaments. And as these guys elongate, and there's these various so-called actin binding proteins that are going to encourage this process, you start getting something like this. You push the plasma membrane out. And this is kind of almost in three dimensions. If you picture this as a cross-section of sort of a three-dimensional kind of ring that rises up. And then it starts kind of folding over a little bit, like so. And that forms a structure known as the fertilization cone. Now, as a matter of fact, I think the textbook has a picture of the fertilization cone in a scanning EM shot, which is actually quite cool. So you see that mass, and it's here again. It's a cell extension that forms sort of a ring that reaches out, and that's because of these growing microfilaments, and it pushes the plasma membrane out. And then kind of starts to partially collapse on itself. And then what's going to happen is we're going to start breaking these microfilaments down, and it causes this cone structure to sort of retract. And as it does so, it starts drawing the sperm head in, actually pushing it through the plasma membrane, and it pulls it in further and further as it collapses. So the egg actually is very important in actually drawing the sperm into the egg. The egg does the dirty work on that. Once the sperm hits the plasma membrane, its job is pretty much done. Now the egg has to do the dirty work. Believe it, right? Isn't that always true? The women generally have to do all the dirty work around the house. Like, I'm a sperm, pull me in, you know. Pull me in. You do all the work. You take out the trash. You pull me into the egg. You know, all that kind of stuff. You know. Yeah, some people are smiling. Been there, done that, right? Okay. Sorry.
So the fertilization cone, like, contracts?
Yeah. Mm-hmm. It seems to be probably a partial contraction, and at the same time, those long microfilaments, you start pulling the subunits off the microfilaments, the active subunits off, so the cone kind of shrinks and starts to collapse because of that. And then the other thing are these proteins are called severing proteins that cut the microfilaments into shorter pieces. Think of, you ever seen those pictures of those, or films, or maybe in real life, those controlled demolitions where they topple a building or a bridge? What do? They generally put the explosive charges on the key structural supports and fire them off, and it cuts like the major support columns and stuff like that, and then the whole building collapses on itself. So you can, in fact, the people that really are good at that, they can topple a 20-story building with about 30 pounds of explosives if you do the real finesse way and stuff. And if you just want to blow a building up, just like, you need a couple tons to topple a building just from a truck bomb or something like that. Just ask a bunch of people in the Middle East. They're very good at that kind of stuff. Yes?
So maybe consider the fertilization cone once it starts to bear, like, very older sperm, or is it,
Yeah.
you know,
Picture it rising up. Let's see if I can do. Let's see if I can make this. Okay. All right. Let's suppose this is a sperm. Here's the plasma membrane. I make a fertilization cone, and it kind of rises up, and then partially collapses over it, and then starts pulling it in, and down it goes. Okay. All right. Okay. So we get that fertilization cone and pulls the sperm in. Now, one thing we see here. The sperm tail is severed and left behind. The tail does not go in. Most of the cytoplasm doesn't go in either. As a matter of fact, at the base of the sperm tail, there's a bunch of mitochondria to power the flagella movement. Those mitochondria are also left behind. Now, occasionally a few sperm mitochondria do get in, and then the egg destroys them. So that's why mitochondrial DNA is maternally inherited. In other words, you get your mitochondrial DNA from your mother. Daddy does not contribute because daddy's mitochondria were left outside the egg cell when you were conceived. And a few that may have gotten in were destroyed by the egg. So there are, for instance, a few mitochondrial diseases that are caused by defects in mitochondrial DNA. And those are inherited only through mother. If mother has it, you'll get it. If daddy has it, you're safe. That, by the way, before they discovered mitochondrial DNA, that baffled geneticists, this maternal inheritance problem for a few weird genes. They couldn't figure out how it was done. And then when they discovered that mitochondria themselves had DNA encoded for a limited number, a few dozen proteins, then they realized, okay, that's your maternal inheritance because the mitochondria from the sperm don't enter in, or the few that do get destroyed. So we're pulling basically the sperm head. Now when you look at mammals, mammals don't really have a fertilization cone. They have a slightly different kind of pattern. So what happens is the sperm actually is kind of on the side, and mammalian sperm is often fairly elongated. Here's binding to the receptors, and then starts causing little kind of blebbing of the egg's plasma membrane, and starts to fuse with the sperm's plasma membrane, and then those fused regions start expanding more and more and contracting a little bit, and then you get something where the membranes kind of fuse, so now you have the sperm head and the nucleus, and the two membranes are fused and gets pulled in. But here again, that also requires the microfilament cytoskeleton. It's a somewhat different process. Most vertebrates and most invertebrates use this fertilization cone trick, but mammals seem to have that kind of membranes merging together and blebbing and stuff like that. So that's a little bit different. Okay, so now we've taken in the sperm nucleus and left very little else that we've taken in from the sperm. And now we also have to deal with the issue of egg activation. We can turn this.